I was just wondering about microbiology phosphorus fixation in the soil and any research that's going on. It seems we look at soil tests regularly and there's enormous phosphorus levels in the soil, but if we don't apply phosphorus we're seeing diminished yields and such. I'm just wondering what your thoughts and any research you know that's going on.
Yes, there is some research going on the microbial release of phosphorus from locked up forms in soil, and the grains industry is funding a couple of projects at the moment to look at that. There has been previous work in the past as well to identify bacteria that release phosphorus, because as the estimates have it that of the fertiliser phosphorus that's added, up to 80% is locked up and remains that way. It doesn't go anywhere, so just it's the phosphorus bank. And so what we need to do is come up with mechanisms for releasing it. So there's some work that's looked at various bacteria that by different mechanisms can release it. So there's enzymes that they produce that can unlock the phosphorus. There's also organic acids that are produced that can unlock that phosphorus as well. There's work that's looking at the role of break crops in cropping situations and whether they can release it through production of organic acids as well. But there is a fair amount of work going on in that space at the moment.
When there's been a soil test has shown a deficiency in the soil or there's a need to add some input, what would your position be on adding that nutrient through the composting process with the compost at the end or independently?
Good question, Tony. I think in principle it makes a lot of sense. In practice, I don't know of any research that has looked at the impact of adding different kinds of fertilisers into compost. So we have to proceed with a certain amount of caution. So, for example, if we decided we wanted to add lime into compost, it probably wouldn't take too much lime to put the compost fire out. Similarly, if we wanted to add urea into a compost, that should only be done really at the very end, and we should make sure that that's well blended through before it's applied. So it makes a lot of sense in terms of reducing the number of applications that need to go out across the paddock, and also in terms of fortifying a compost, like I was saying before about the need to supplement compost with inorganic nutrients. Putting them through the compost seems a very sensible thing to do if it's late in the composting and we're using it simply to do 2 application jobs at the one time. There may be some advantage in putting some elements through the compost early on, for example some phosphorus compounds, to have that material converted into a more organic form before it. But again, without research on that, we can't say what's best.
So really interesting that there might be a lot of phosphorus in the system but not available. So where the rubber hits the road, I'm kind of interested in your opinions on what you might expect some of the management actions might be to unlock that phosphorus. And I know that each soil's going to be different, each constraint, but just some general management actions that you might advise a farmer if they've got high unavailable P in their system, or even just high available P but it's not being taken up. What other kinds of management actions that you would recommend?
I'll just start with a first little input there. I'd be starting perhaps from the chemistry to start with, just to look at the pH, because we know that around about pH of 5, for instance, the phosphate will be locked up as iron aluminium phosphates. The same turned up in alkaline soils, you're going to get that fixed as calcium phosphate. So pH adjustment is one of the first things I'd look at. Then we go back to the soil structure. If we want an environment for the phosphorus solubilising microorganisms, as they call them, then they've got to create an environment that's conducive to them existing there. So that might be looking at, for instance, the soil structure. Are there compaction issues? What's organic matter content? Because they need that food source. So we're looking at that whole cycle, if you like, of the physical, the chemical aspects of a soil. to make sure that that environment is suitable for those organisms to actually work.
What sort of questions were you being asked in your role with FERT this morning that you'd like to share?
Two key areas, I guess, is where can I find credible information, and you know, what constitutes good advice, I guess, at the end of the day. And I guess that's where FERTCare and the FERTCare Accredited Advisor program has a part to play in terms of making sure that the advice that's given is based on accepted science, and there's evidence for the claims that are made. And I guess associated with that is you know, having good information about the description of the products that you're using. And I think generally as a industry, particularly some of the inorganic forms in the compost area, we've probably got a little way to go in terms of having a uniform standard for that. But yeah, that's one area.
Durgan, in Gippsland, a lot of our soil tests are running at 10 to 12% organic matter. So really we're trying to break that down to get it back to around about the 5. Does compost work better in low soils, like 1 to 2% organic matter, sandy soils that need building?
Firstly, if your soils are very low in carbon and they're deficient in carbon, in other words, they should be and could be holding more carbon, if you put the compost on to those soils, then we should see a response from that compost, either in the form of, say, increased pasture production or an improvement in soil condition. What's interesting about your question is some of the observations that we've seen in dairying. And I remember when I first started working with dairy farmers who were using compost, you know, I did think, are we bringing coals to Newcastle here by applying compost to dairy farms? Because dairy farms traditionally have the highest levels of soil carbon pretty much of, you know, across the various industries. And I don't think we have research that really answers the question of, you know, what's the real value of adding compost to high carbon soils. But I'd be interested in Pauline's take on this, because one of the speculations that I've had, and it is just a speculation, is that some of the reports from dairy farmers about how their soil condition has declined over time, and also how the response to nitrogen has declined over time, is suggesting to me that high rates of urea in particular, I think, or nitrogen in particular, is possibly shutting down or reducing the amount of biological activity in the soil, or the activity required to maintain soil condition. Now that's a little bit of a paradoxical statement, because we've got lots of carbon and we think, well, if we have lots of carbon in there, surely that's enough. But of course, I liken carbon to money in an economy. If all our money is in the form of gold that's sitting in a vault somewhere and is not circulating, then it's not really doing our economy any good. And similarly, we might have high carbon in the soil, but if it's not cycling, then it's not doing the system very much good. So we want our carbon to be cycling. We want it to be breaking down. We want the carbon cycle, in other words, to be functioning as well as we can, so that we have capture, we have release, as you know, as intended by the cycle. So my speculation is, did we have kind of frozen assets of carbon in dairy soils because of management practices? And is the reduction in the amount of inorganic nitrogen going into dairy farms, plus the stimulation of adding compost onto dairy farms, is that re-establishing the carbon cycle? Thoughts, Pauline?
Well said, Declan. I think that's probably right. I think a really simple observation that we've made is that in sandy light soils, you tend to see stronger responses to adding organic matter in terms of microbial turnover than, say, a heavier clay soil. That's just, you know, how they work. I think if you've got reasonably high levels of organic matter, and it sounds like you have, you know, a lot of our cropping soils have got less than 1%, so 10% is, that always amazes me. But I think I would be looking at whether you've got a microbially active community there, and if it doesn't appear to be very active, and you know there are ways of measuring that. So if it's not turning over, I would be asking what's preventing, what's constraining that system from turning over. I suspect it's a dairy system, so is it a dairy farm? Do you, oh okay, it's beef, okay. You add fertilisers, do you add fertiliser? Yeah. Yeah, okay. My guess, what's the pH? Yeah, so if you've got, say, pretty high organic matter and you're in a high sort of fertiliser input regime, my guess would be that the system is acidifying. It's just and you're taking product off, maybe you're cutting hay, I don't know. But if the system is acidifying, the microbial turnover will be slowing down, will be running down. So, yeah, I think that's, in a high organic matter system, you've got to look at whether you've got good turnover and what's constraining that turnover, and perhaps pH is one of the constraints.
In a situation in an orchard, we're continually spraying pesticides so we can have a crop to sell, and I would imagine that would be having an effect on the microbe activity. It's killing things in the trees, I'm assuming it's killing things as it leaches into the soil. If that's the case, have you got any suggested solutions to mitigate that issue?
It is likely that, particularly where there are residual amounts of pesticide in the soil starting to build up, and that happens when there's extended periods of dry, so they tend to, you get the residual effects. Then it's been shown for some pesticides that you do see, I guess, a detrimental impact on nitrogen cycle and rhizobia and a few other groups. And that's where you've got residual. If the system is, if you've got a lot of moisture, a lot of carbon in that system, then you'll have quite a bit of microbial churn going on anyway. So they'll be able to, they just see pesticide as another carbon molecule. And they see it as food, therefore. So they will break it down, but if it builds up too much, then I think we run into problems. So are there strategies? It's hard to know. I'm not familiar with the orchard industry, but I would imagine if you could keep the chemical away from the soil surface as much as possible.
IPM strategies.
IPM strategies, yeah, they're good. But if you have a lot of contact between the microbes in the soil and the chemical, you may have some problems. But I think the biggest issue is with the residual effects.
We've been trialling compost. We have a control row, a fine mulch row, and a coarse mulch row. In all those rows, we have gypsum blocks and temperature sensors, and they're doing pretty much what everyone would expect them to do. The control row is drier than the other two rows. The coarse compost row is actually, I think, the best. The interesting thing was the soil temperature. The soil temperature at the moment doesn't drop below 20 degrees on the two composted rows. On the control row, it drops down to about 18, which doesn't seem much. But our experience has shown that it's instead of the graph being low, high, day, night, the composted rows are more of a curve, so it doesn't get as hot, doesn't get as cold, doesn't get as dry, and doesn't get as wet. So, but that's what the trial's about. We don't know if that's a good thing or a bad thing. It could be that vines want it to get hot and cold.
Just a quick one. Our colleagues in New South Wales have been using a phosphofluoroacetase enzyme test to measure biological activity, and they see that as being a possibility. I just wondered what you think about testing enzyme activity, just to give us an idea of the biological activity in a compost or soil sample.
Yes, enzymes are a good way of determining what's happening in your soil, what functions are being carried out. Again, you can, there are some traditional ways of doing that, or you can do PCR and look for enzymes, or you can, look at, the whole genome and look for enzymes as well. They're very good. I wouldn't rely on one measure. So if it's the fluorescein diacetate hydrolysis, it measures all hydrolases, so it's a fairly generic measure, and it's like biomass, so it'll give you and it's like Solvita, it will give you a general measure of activity. I think, and it's all about availability, but I think we've got to move down the path of being more specific about the enzymes, or get a whole suite of enzymes that we measure, because each of those enzymes are measuring different things in our system. So it will give us a more complete picture if we can develop measures that capture a whole range of different processes. So phosphatases for dissolving phosphorus, lipases for dissolving, or lacases for dissolving, larger carbon compounds, nitrogenases for nitrogen fixation. So there's a whole family of enzymes we should be looking at. And methodologies are developing so rapidly now that we can simultaneously measure a whole range of things together. It comes back to what question you want answered, whether you are most interested in what's happening in your nitrogen cycle, whether you're losing nitrogen quickly. So you can measure a particular enzyme to tell you that. So it does depend on the question you're asking. If you're interested in whether you're getting breakdown of a particular, say your compost, you would measure a different enzyme set. So enzymes are good is the answer to that question. But if we can develop tests that measure a whole family of enzymes, that would be good. Or alternatively, know what our question is, so we can target the right enzyme.
I work with EnviroTurf, and we actually do quite a lot of work with compost tea, and I've just been sitting here waiting, but there hasn't seemed to have been any mention of that during the talk so far. And we've got some tremendous results. We work with golf clubs, wineries, vegetable growers, and the diversity of the compost tea going up. We've really had some terrific results, but I was just interested to hear what your comments were on that.
Compost tea, as I understand, they're made up of humic acids and fulvic acids, and they're a readily available source of carbon for a lot of microbes that are in the soil. So I would see that the way they work would be to prime quickly the microbial community that's responsive to ready forms of carbon, if you like. And so having that compost tea will, I guess, quicken turnover of nutrients that might be locked up in the soil. So it's, I guess it's a stimulant for the biology that's present in those systems. And when they're, I guess you're likely to see responses in situations where there's perhaps low levels of carbon. So perhaps in Western Australia you might see that, where there's less than 1%. So, but yeah, look, I guess I see them as having a role in stimulating turnover of nutrients that might otherwise be locked up. But I don't know of any, extensive studies to show exactly how they work.